Dear ladies and gentlemen, I feel honored to speak here today. In this topic, I will talk about paracrine factors released by non-irradiated and irradiated human peripheral white blood cells. First of all, I would like to thank all those people who helped me to finalize this project. In the center of my presentation are these cells. This is a monocyte, or a picture of an electron microscopy of a monocyte. And I will talk mainly about PBMCs, about peripheral blood mononuclear cells, and their factors that they release in vitro. Since 2009, it's known that if you culture white blood cells for 24 hours and you irradiate these cells, that these cells release factors. These factors can be lyophilized and you receive a powder. If you use this powder and inject this powder into animals suffering from acute myocardial infarction, these animals show reduced scar formation and improved functional parameters, as shown here in this rodent model of acute myocardial infarction. We further elucidated the effects of this powder, named APOSEC, that's an acronym for apoptotic cell secretome, and were able to evidence that the application of APOSEC attenuates area of necrosis in large animal models. As you can see here, these are APOSEC treated animals in comparison to control treated animals, and that APOSEC improves the myocardial infarction at 3 weeks after primary injury. In 2013, it was published that the application of APOSEC attenuates the severity of myocarditis in an experimental model of myocarditis, and members of our lab were able to show that these factors augment wound healing. You see here pictures of marine wounds at day 0 after injury and day 7 after injury. If you use this powder and resuspend this powder in a gel and applicate this gel on the wounds, these wounds close more rapidly in comparison to control-treated animals. Furthermore, the wounds show a more rapid reepithelialization. As you can see here, this is the newly formed epidermis in APOSEC or in secretome treated animals, whereas control treated animals did not evidence a comparable thickness of the epidermis. And it has been shown that the application of APOSEC attenuates ischemic injury following stop of blood flow in a rodent model of experimental stroke. Okay, and we know that these factors have pleiotrope effects on different cell types and different organs, and we know that these factors are released by stressed human PBMCs. However, we do not actually know what's really present in these suspension. What are the biological active components present in this powder? Therefore, we performed this study and aimed to investigate different biological classes present in APOSEC or in the secretome of stressed leukocyte. We focused on these 4 classes of biological components: first, proteins, then lipids, microparticles, and exosomes. This slide illustrates our experimental setup. We first separated PBMCs from healthy donors. We either irradiated them with 60 gray or non-irradiated them and cultivated these cells for 24 hours. Thereafter, we first isolated the cell pellet, used the cell pellet to obtain total RNA content, used this RNA for microarray analysis. By doing so, we tested 4 genes that code for secretory proteins, and then we investigated the biological function of these genes coding for secretory proteins. And on the other hand, we collected the supernatant of these cells containing all these biological components, and then we isolated lipids, exosomes, microparticles, and soluble proteins of these supernatant and analyzed these biological components and used them for in vitro assays. First, proteins. We know since 2011 that the secretome of irradiated white blood cells contains many proteins associated with angiogenesis, with chemokine signaling, with proliferation. You see here membrane arrays with pro-angiogenetic proteins, and for example, the protein MCP1 or IL-8 is significantly enriched in the secretome of irradiated PBMC. Here's a table showing all those pro-angiogenetic proteins or cytokines present in the supernatant. However, these techniques are limited to a number of proteins. You cannot detect the whole secretome by once. Therefore, we decided to use the microarray to identify genes coding for secretory proteins. By doing so, we identified 179 genes coding for secretory proteins in non-irradiated PBMCs. On the other hand, we found 213 genes in irradiated PBMCs that code for secreted proteins, indicating that irradiation induces the expression of genes coding for secretory proteins. And then we questioned, okay, we know that these genes are present in irradiated cells. What's the biological function of these cells? And we found by a GO term analysis that these genes were associated with blood vessel development, with the positive regulation of angiogenesis, with wound healing, with hemostasis, with the regulation of coagulation, and last but not least, with leukocyte migration. This is in line with previous data showing that the secretome of these cells enhances wound healing, attenuates platelet aggregation, and modulates immune function. We then performed validation experiments using ELISA, and were able to show that all these proteins here were present on protein level in the supernatant of irradiated, given in red, and in part also present in the supernatant of non-irradiated white blood cells. The second point were lipids. Why lipids? It's well known in the literature that lipids, especially oxidized lipids, induce or promote angiogenesis. You see here oxidized lipids that bind to cell surface receptors, which activate intracellular signal cascades, which lead to the release of pro-angiogenetic proteins such as IL-8, MCP1, and other chemokines. And it has been shown that the application of selected oxidized phospholipids induces sprouting of blood vessels or of UVECs, as you can see here, whereas the application of non-oxidized lipids does not induce the proliferation and blood vessel formation of these cells in a comparable manner. We first performed simple types of analysis, such as a thin layer chromatography. By doing so, we were able to see that both non-irradiated and the secretome of irradiated PBMCs contained several classes of lipids, for example, phospholipids, cholesterol, or triglycerides. If you compare these bands with the bands present in keratinocytes, you can clearly see that PBMCs secrete less classes of lipids in comparison to keratinocytes. By performing an image J quantification, we were able to show that ionizing radiation induces the release of nearly all classes of lipids. Thereafter, we performed a high-pressure lipid chromatography, especially focusing on the presence of oxidized phospholipids. Why oxidized phospholipids? Because it's known that especially these class of lipids induces angiogenesis. You can see here on the left side of the slide precursor forms, non-oxidized phospholipids. And what can you see? That basically the concentration of these non-oxidized lipids is comparable between irradiated and non-irradiated PBMCs. Whereas the oxidized form of these phospholipids is significantly enriched in the secretome of irradiated PBMC, suggesting that irradiation induces the oxidation and spillage of oxidized phospholipids. Then we focused on microparticles and exosomes. Extracellular vesicles can be classified based mainly on their size. The smallest particles are exosomes. Exosomes had a diameter between 30 to 100 nanometer, whereas microparticles, are also known as microvesicles, have a diameter between 100 to 1 micrometer. What's especially with exosomes, why are they so important and so highly discussed nowadays? Because exosomes are produced intracellularly in so-called multivesicular bodies. They can be stored inside the cell, and upon activation these particles can be released into the extracellular space, as you can see here. And especially exosomes have been investigated in detail in the field of regenerative medicine. It has been shown that exosomes stimulate the wound closure or the migration of HUVECs in vitro, as you can see here. Cells stimulated with medium alone, whereas cells stimulated with medium and exosomes, that these cells migrate more rapidly. And exosomes has been shown to attenuate myocardial infarction in a rodent model of acute myocardial infarction, whereas exosomes from fibroblasts did not attenuate the infarct in the same or comparison to exosomes released from mesenchymal stem cells. What can we find? What were we able to show? First, we were able to show that the supernatant of non-irradiated and irradiated PBMCs contained microparticles. However, ionizing radiation induces the release of both small and large microparticles. As you can see here, these are small microparticles in non-irradiated samples, whereas these are microparticles in irradiated samples. However, I have to add that microparticles were not present in the GMP-produced APOSEC. What's the GMP-produced APOSEC? GMP means that the APOSEC, the secretome, is viral clearance. It's not possible that this contains any viruses anymore. Exosomes. What can we see? Here you see exosomes from non-irradiated and irradiated PBMC. However, we found that irradiation induces the release of exosomes, and that the exosomes contained more proteins in comparison to non-irradiated PBMC, in comparison to exosomes derived from non-irradiated PBMC. That is shown here. We've performed silver staining of proteins present in exosomes and were able to show that exosomes from irradiated PBMC has a different quality of proteins in comparison to exosomes derived from non-irradiated PBMC. Thereafter, we checked whether we could really separate exosomes and not cell garbage. And as you can see here, exosomes from non-irradiated and irradiated PBMC were both positive for typical exosome markers such as CD63 and CD9. We furthermore performed a two-dimensional Deutsch analysis. In this slide you see proteins, exosomal proteins of irradiated sample stained with a red dye, whereas exosomes from non-irradiated PBMC were stained with a green dye. Yellow spots show protein spots that were present in both in the exosomes of non-irradiated and irradiated samples, whereas these red dots, here, here, and here, are proteins that were only present in irradiated samples or in exosomes derived from irradiated PBMCs, indicating that ionizing radiation induces changes in the protein content of exosomes. To conclude, we were able to show that proteins are present in the supernatant of irradiated samples, that lipids and different classes of extracellular vesicles are present in the APOSEC. And what is known since 2009 is that the secretome of these cells augments the expression of different pro-angiogenetic proteins or genes. For example, if you cultivate fibroblasts with the secretome of irradiated PBMCs, these fibroblasts induce or upregulate the expression of VEGF and CXCL8 or IL-8. And it's well known that this secretome enhances wound healing, as you can see here, in a more thickness of the epidermis. And furthermore, we were able to show that APOSEC, or the secretome of stressed leukocytes, enhances the migration of fibroblasts and keratinocytes. And we questioned in this study, could we identify the biological active component present in APOSEC that is responsible for this effect? And therefore we first performed some in vitro studies showing that the supernatant alone, the whole suspension, induces CXCL1 and CXCL8 in fibroblasts, as you can see here. If you separate the different classes, this effect was also seen, but only for exosomes and the protein fraction, indicating that exosomes and proteins induce the expression of pro-angiogenetic proteins in fibroblasts, for example CXCL1 and CXCL8. We also performed evaluation of the cell of the supernatant of these fibroblasts, and we were able to show that exosomes derived from irradiated and non-irradiated PBMC induce the spillage of CXCL1 and CXCL8 in the supernatant of fibroblasts. Thereafter we questioned, okay, what's the biological active component that activates fibroblasts or keratinocytes to more rapidly close wounds? We performed these scratch assays. Here you can see the initial scar. This is the initial scratch, or the picture of the initial scratch taken immediately after injury. We stimulated these cells with either control medium or exosomes derived from non-irradiated or irradiated PBMC, and after 13 hours we measured the wound area. This is the planimetric examination of these photographs, indicating that both exosomes from irradiated and non-irradiated PBMC augment the migration of fibroblasts and keratinocytes. And we also found that the protein fraction is able to induce cell migration, whereas microparticles or lipids did not augment the migration of fibroblasts or keratinocytes. To sum it up, or to draw a conclusion, we show that extracellular stimuli such as irradiation or ex vivo cultivation stresses leukocytes and induces the release of exosomes, microparticles, proteins, and lipids. Ionizing radiation induces especially the release of oxidized phospholipids. And we were able to show in a selected set of in vitro experiments that proteins and exosomes are the biological active components that induce migration and the expression of pro-angiogenic proteins in fibroblasts and keratinocytes. To conclude, ionizing radiation leads to the enhanced release of secretory protein, of oxidized phospholipids, and of extracellular vesicles. APOSEC, produced according to the GMP guidelines, does not contain any microparticles. And we were able to show that exosomes and proteins are the biological active components in our in vitro assays. What's the outlook? We believe that exosomes and proteins are promising targets for cell-free therapies in the field of regenerative medicine. We speculate that the application of these biological classes results in pleiotrope in vivo effects. We are currently not able to identify the biological active components for all of our in vitro and in vivo settings. But we propose that APOSEC, or the application of cell secretome, induces systemic effects, and we're currently investigating these effects in further in vivo studies. Thank you for your attention.